As I'm sure you can tell, I'm a very serious person. I wore a t-shirt that I got printed at Zazzle. It says, proud to be a GMD, that's Guardians of Microbial Diversity. And my kids, 5 and 7, drew what they thought of as microbial diversity. I don't quite know what any of them are, but they even crossed out some of the words. I don't know what that was about. So as you will see in my talk, I'm also very, very serious when I give talks. So thanks, Jeff. Thanks for everyone coming to a evening talk before the drinks. I guess that's the way to keep people at least somewhat. I've done the after dinner talks after the drinks, which is really a disaster. So as many of you know, I'm kind of obsessed with the history of this meeting. I've been coming for many years, not the whole way, but many, many years. If you're interested in some of the past history, I've posted scans of many of the programs from the last 10 or so of these meetings. I made a tiny URL link for them, so it's easy for people to find at tinyurl.com/LAMG12. That's also the Twitter hashtag for the meeting for all the people posting to Twitter. It's LAMG12. And there's all sorts of stuff there, notes from previous meetings, previous blog posts, my hand notes, and I'm sure there's really offensive stuff somewhere in there, so please forgive me. But really the key to the history of these meetings is captured, and I'm going to need your help with this for this year, in quotes from previous years. So when you hear something good, like we just heard a minute ago and I wrote down, Microbial. Please record those. We need a record of these good quotes. And I'm going to go through some of them here, not telling you who said them or when they were, but someone in one of their talks referred to the space-time continuum of genes and genomes. Microbes not only have a lot of sex, they have a lot of weird sex. That must have been Rosie Redfield, right? I mean, gene sequences are the wormhole that allows one to tunnel into the past. Yeah, everybody's nodding. I have no idea what that means. This is how you do metagenomics on $50, and that's Canadian dollars. This shows the quality, the precision of the science at this meeting. The human guts are a real milieu of stuff. Antibiotics do not kill things, they corrupt them. And I'm not done. There comes a point in life when you have to bring chemists into the picture. I disagree with this statement, by the way. That really is true. I'm not even going to read it. If I have time, I will tell you about a dream. And that was in a talk, not at the bar. Another thing you need to know, actually you don't need to know any of it. Lance, I have been influenced by Fisher-Price throughout my life. If you have the pleasure of seeing Lance show all of his Fisher-Price toys in his talks. This is going to be ironic coming from someone who studies circumcision. Lance. And we will bring out the unused cheese from yesterday. That must have been Jeff. A paper came out next year. The people here are really brilliant. It takes 1,000 nanobiologists to make one microbiologist. And last but not least, in an engineering sense, the vagina is a simple plug flow reactor. So now you know what I'm all about now. Please record quotes, post them to Twitter unless they're Rich Roberts telling a bad story. Or send them to me and I will keep an archive of them. All right, so now I'm sorry, I'm actually going to give a talk. So what I want to talk about today, and really there are no more quotes, What I want to talk about today is focusing in on taking a phylogenetic, or you can call it phylogenomic if you integrate genomic data, although I'm kind of hate omic words, even the one I invented, phylogenomics, so I probably should have crossed this out from my slide. A phylogenetic approach to studying microbial diversity, and I want to talk about, I've been doing this pretty much for my whole career, and I want to talk about some new developments, mostly in terms of methodological developments that have either come from my group or from collaborators, to sort of give you prime the pump here, so to speak, for the rest of the meeting, where I hope that people think about explicitly phylogeny and the history of organisms and of genes when they're looking at some of the data that people will be presenting. And the first thing I want to talk about, which I assume everyone or virtually everyone here is familiar with the general principle, is phylotyping. So phylotyping is developed, originally by Norm Pace and colleagues, where you go to an environmental sample, you clone out originally ribosomal RNA genes and still predominantly ribosomal RNA genes, sequence them, take the sequences, you build a sequence alignment with them, and you can do lots of analysis from that sequence alignment. But phylotyping explicitly really is supposed to refer to phylogenetic analysis from that sequence alignment. So you can build a tree and try and understand what the uncharacterized organisms are, that is, who is out there, from building a phylogenetic tree of the sequences and comparing the sequences that you get from an environmental sample to uncharacterized organisms. And that tree is really important because it tells the history of the relationships among the organisms. And it's explicitly different than a grouping of things by their similarity to each other. If you just look at the similarity of organisms or genes to each other, you may be misled in cases, for example, of unequal rates of evolution in different taxa, where the level of similarity will not reflect the evolutionary history of the organisms or genes. So you build this evolutionary tree, you try and classify the uncharacterized sequences by how they relate either to each other or to characterized organisms. You can deviate from this general plan, and a lot of people do this as we get more and more sequence data, more and more people are doing this, where you take the sequence alignment or even the sequences directly and cluster them, compare them to each other, and try and identify which ones are really, really similar to each other, and pull out the dreaded OTUs from that clustering, some groupings that correspond to species or some taxonomic group that you think all the entities in that group are closely related to each other. And the entities in different groups are distinct from each other. And you can come up with a list of the OTUs from this clustering without ever doing a phylogenetic analysis. However, it's been shown in a variety of papers in a variety of ways that if you layer on top of that OTU clustering the phylogenetic relatedness of the OTUs, you can do a lot with that data. So for example, this is the basis behind things like unifrac analysis, where you compare two different communities by not the list of OTUs in those communities, but by the phylogenetic relatedness of the organisms in those communities. So you can also go through, of course, as I implied before, rather than going to OTUs directly, and build your tree first, and then identify either OTUs from that tree, or actually directly do phylogenetic analysis without ever identifying OTUs. So there's this sort of suite of ways that people take ribosomal RNA data and end up with either a tree or OTUs or a tree of OTUs that they classify. And there's been, 25 plus years of people using phylogenetic approaches to analyze this type of data and shown that it has been useful in a variety of ways. Identifying novel groups, identifying clades, looking at rates of change in organisms. If you have different genes, you can look at lateral gene transfer, convergent evolution, questions of phylogenetic diversity, etc. I'm not going to go into any of these in real detail, but just want to again emphasize that phylogenetic approaches are somewhat distinct from just getting a list of the taxa that are present in a particular sample, because they tell you how closely related particular taxa are to each other. So what I want to talk about instead is not sort of the history of this and justification for it, but what's really new in the last few years in phylotyping. And, as I'm sure everybody here appreciates, the new thing in phylotyping, or one of them, the first one that I'm going to talk about, is what you can get by cranking out data from one of the new toy wonderful sequencing machines that we all use, or that many of us use, where you can generate just literally absurd amounts of data for a trivial cost compared to what you could have done 3 years ago, 5 years ago, 10 years ago. So obviously, if you're doing ribosomal RNA PCR, you can generate more PCR products, but that in and of itself isn't necessarily the key thing. What many people want to do is deeper sequencing of individual samples. That is, you can get the tail end of the relative abundance curve, and if you're interested in a particular sample, you might get to what we have been calling the rare biosphere, many people in the community, and maybe get more accurate relative abundance estimates of even the abundant organisms by sampling more deeply. What I think many people here will talk about or show posters about is taking the sequence data that you can generate, and instead of generating deeper sequencing, generating data from more samples, time series, spatially diverse samples, fine scale sampling, etc., where you can barcode from ribosomal RNA PCR, add barcodes to each of your samples, and pool together hundreds to thousands to possibly tens of thousands of samples into a single run of one of the sequencing machines, and thereby generate data that we have never seen the likes of which for microbes before. I mean, people have collected this type of data for plants and animals from thousands and thousands of sites. It's hard, but people have done it. But for microorganisms, we generally don't have this type of diversity data from across a wide number of samples. So this is just an example of what can be done. This is a paper that I was a co-author on with Jen Hughes-Martini and a few other people from Jen's group, where she was interested in beta diversity. And when I was at Tiger, prior to moving to UC Davis, I helped her sequence with Sanger sequencing. People know what that is? Should I explain? With Sanger sequencing, something like 10,000 ribosomal RNA sequences. I mean, that's like 5 cents now with Illumina sequencing or something. And she, in analyzing this data and in planning the experiment, was able to generate really interesting findings related to biogeography of organisms and beta diversity across the planet. And we thought, you know, this is amazingly deep. We had, you know, 5 sites that are closely spaced next to each other, and in total, like 48 sites across the world. And now you have things like the Earth Microbiome Project, where I don't think anybody's going to talk about it here, but there, Jack Gilbert and colleagues are literally talking about generating data from millions of samples across the globe instead of 48. And you can do this, again, because of the possibility of barcoding and pulling together samples. And what we're going to be able to do is generate really a biogeography of the planet, at least at some level. And if we choose this well, we can do good biogeography. And I would argue that even though, and I'll come back to this in a minute, even though you may feel like you're drowning in data, it's still useful to take a phylogenetic approach to analyzing that data, even if you think, oh my God, I can't build trees with this data because there's too much of it. So you can do things, for example, with that data. I have a colleague, Josh Ladow, who's in Katie Pollard's lab, who's been working on taking biogeographical data and trying to actually develop range maps for microbes. There are a few other people, Noah Fier and colleagues have been starting to do this, where, you know, sort of like in a bird field guide where you have a range map of the birds. We don't really have that for environmental microbes out there. We have 10 samples from many places, but this is trying to take a modeling approach to develop range maps to organisms. I made this argument a couple of weeks ago, and I thought I'd just go through it again. I mean, 5 years ago, making this argument would have been absurd to talk about DNA-based studies of microbes in the Mississippi River. It's 2,320 miles long. If you want to do 1 site per mile, 3 samples per site, that's not a lot of redundancy, but let's pretend like that's good. That's 6,960 samples. With barcoding, you can generate, if you get really good runs with a MiSeq or HiSeq, you know, something like 4,300 sequences per sample in a single MiSeq run for those 7,000 samples, or 862,000 sequences per sample if you do it in a full HiSeq 2,000 run that all works perfectly and everything is high quality. We're going to ignore all the problems with that. But even so, I mean, literally, you can now imagine going out for $5,000, $10,000, whatever, creating a transect of a site that you're interested in with DNA-based studies of microbes on a scale with which no one had thought about previously. It's what, as I assume you will hear in the human microbiome sessions, what many people have been doing, in essence, creating a biogeographical map of the human body with the low-cost sequencing. We can now do that in any environment. So what's new in phylotyping? What else is new? Now, obviously, in the last 10 years, one of the big things that's been new is metagenomics. And as sequencing gets cheaper and cheaper, people are not going to just go do ribosomal RNA PCR from an environmental sample, but they're going to do metagenomics from those environmental samples. And obviously, you get fewer sequences per gene that you're interested in. Let's say you were interested in some functional gene, but you get sample of all the genes in your sample rather than just the ribosomal RNA genes. And you can go to environments and do metagenomics just like you would do with ribosomal RNA surveys. Now, it turns out that it's a little trickier, a little more complicated, but you can do the same general approach, phylotyping, with metagenomic data that people have been doing for years with ribosomal RNA PCR data. You can take metagenomic data, cluster sets of sequences that you're interested in, compare them to each other, build OTUs. You can build phylogenetic trees of the sequences that you're interested in and identify OTUs, or just do phylogeny and then take all of the benefits that come from phylogeny, such as doing things like unifrac or phylogenetic ecology or looking at rates and modes of evolution, and do it for all the genes in a sample as opposed to for just ribosomal RNA in the sample. So that's the fundamental difference, is that you can do this for all of the genes in the sample and get maybe a different picture about the biogeography or biodiversity of organisms than was available for ribosomal RNA. Or if you work with organisms that were not amenable to ribosomal RNA PCR, what are those small things for us that they have like a code on them? I mean, so viruses have been ignored by the ribosomal RNA PCR studies, but we can now do phylotyping of viruses, as many people have been doing for the last 4 or 5 years, because of random shotgun metagenomics. So what I want to talk about now is what are the challenges with trying to do this. So let's just assume we're going to generate either billions of sequences or sequences from metagenomics, and now we're going to try and do phylotyping from those samples. Well, one of the big problems, especially if you get your data from metagenomics, is that if you generate a sequence alignment of your gene compared to full-length data mostly from sequence genomes, which is where we're getting most of the full-length sequence data, you get a sequence alignment where your environmental data might be a fragment at the left-hand of the gene, or at the right hand of the gene, or in the middle of the gene, and some of those sequences might not even overlap with each other. So how are we going to build a phylogenetic tree with traditional methods that rely upon a multiple sequence alignment without any gaps in it with metagenomic data? This has been somewhat of a challenge for many people in analyzing large data sets to figure out exactly how to do this. There have been multiple approaches that have been developed, and what I want to do is take you through sort of the philosophy and examples of each of them. So the first that you can do, which many people still do today, is to treat each sequence as an island, in essence. You build a tree of reference sequences, full-length sequences that you have from genomes, and then you take each individual metagenomic sequence and you build a tree of it relative to the reference data. So here you pluck out an individual sequence and you build a tree of that one environmental sequence relative to the reference data. And then you build a tree of the next sequence relative to the reference data and to the next sequence relative to the reference data, and so on. And how do you do this for billions of sequences? Well, you need to start to automate virtually everything. Everybody who does genomics now is becoming more and more familiar with this. We developed a pipeline. You almost certainly don't want to use it anymore because it can't handle millions of sequences, but we developed a pipeline a few years ago to automate this sequence as an island approach to analyzing ribosomal RNA data and building a phylogenetic tree of it called STAP. There are many other pipelines out there that do things like this. We've also built pipelines in my lab. This one was developed by Martin Wu a few years ago called AMPHORA that will do the same type of thing, take a sequence one at a time, build a tree of it relative to reference data, but it will do this with protein sequences as opposed to with ribosomal RNA sequences. And we selected a set of 31 phylogenetic marker genes, protein coding genes that seem to have good properties that would allow them to robustly build phylogenetic trees of the organisms from which those sequences came. And built tools around automating the construction of phylogenetic trees of these sequences using alignments from hidden Markov models and pre-computed masks for the sequence alignments so that you didn't have to pull out regions that were poorly aligned from these sequences. And you can do this with this AMPHORA program and build trees of each individual protein coding sequence, these 31 markers. And you can then scan through data and you get phylogenetic types, phylotypes, for a suite of genes in addition to ribosomal RNA. And even for organisms where you have ribosomal RNA genes, this can be beneficial, and I'll come back to this in a little bit, for example, because copy number of ribosomal RNA genes varies significantly between taxa. And if you want to estimate relative abundance of organisms from the copy number that you see of sequences, and the copy number of the gene in the genome varies a lot, you're going to misestimate relative abundance of organisms without knowing that information. Many of these protein coding genes have much lower variance in copy number than ribosomal RNA between organisms and provide a better way of estimating relative abundance. They also tend to have higher sequence variation among closely related organisms because of third codon position variation that ribosomal RNA does not have. So they allow you to distinguish a finer scale resolution of the phylogeny of organisms than ribosomal RNA. So an alternative approach from this sequences and island approach is to try and analyze some of the metagenomic sequences, not just relative to the reference, but relative to each other. And one of the approaches that has been taken to doing this is to take a sequence alignment and identify a core region of that alignment where many of your metagenomic sequences overlap with each other in that core region of the alignment, throw away everything else, and then build trees from that. That's what I did in analysis of the Venter Sargassov C data in the original Venter metagenomic paper. And you can scan through ribosomal RNA data from metagenomics, build trees just like this, and now compare all of the sequences to each other. You can build trees with protein coding genes as well. I always pick REC-A as my first protein coding gene. Many people know that I'm obsessed with REC-A. And build trees and do the phylogenetic classification again, but now be comparing the sequences to each other as well as to the reference data. And this is getting to be more and more important as we deeply sample environments but don't have genomes from reference organisms that are closely related to the taxa and the environments that you're interested in. You need to compare the environmental sequences to each other, not just to the references. This is, again, how many software packages work. This is what basically people do when they analyze ribosomal RNA PCR data in QIIME, or when you analyze data in Mothur, or when you take metagenomic data and analyze them in certain similar ways. That's what people are trying to do, find a core region, align it, and build a tree from that, and then do analysis. Just as a brief aside, I wanted to mention this. A graduate student, previous graduate student in my lab, Amber Hartman, developed this software pipeline called Waters, which is for analyzing ribosomal RNA data, and it's what's called a scientific workflow system. It's embedded within the Kepler scientific workflow package. And this is not the easiest to use system on the planet, but it's going to be really more and more important in the future as everybody starts to do bioinformatics. What scientific workflows do is basically record everything that you do with the data that you're looking at. They have full provenance of every setting, every input and output, flow of data, conversion into different data formats, recording of the operating system that you're using, recording of every sort of feature of the data, and that's going to be more and more important for sort of record keeping as we basically become a data-driven world. You can make your workflow available to people, record everything you did in one particular workflow, and people will be able to, at least in theory, reproduce what you did a little bit more than in many cases in bioinformatics. So I've already mentioned this a couple of times. There's one major problem with relying upon ribosomal RNA where phylogeny can actually help us, and that's this copy number variation. And for many years, people have been looking at the copy number variation in ribosomal RNA and saying, there seems to be some biological consistency with the copy number. There's been a lot of papers about organisms with more copies grow more rapidly than organisms with fewer copies. There have been a lot of studies about the ecological context of organisms that have more copies. But there's also phylogenetic consistency in the copy number. And Stephen Kembel, who was a postdoc in Jessica Green's lab and is now moving on to a faculty position, worked on this with Martin Wu, who used to be in my lab, to do to track the phylogeny, the phylogenetic history of copy number. So that's what's shown in this figure. There's a paper in press in PLOS Computational Biology that Steve's the first author on, where this is a phylogenetic tree of organisms, and then the black bars are the copy number of ribosomal RNA for those organisms. And there is some phylogenetic consistency to this copy number. What that means is that you can correct for the copy number over or underestimate that you get from ribosomal RNA sequence data by looking at where you are in the phylogeny and in the copy number pattern in the phylogeny, and get a corrected estimate of relative abundance of organisms using the phylogenetic history of copy number to make your estimate of what the copy number is going to be in a new organism. It is not perfect. Copy number varies in ways that are not related to the phylogeny or not predictable right now, but it is better than not using this correction. So a third, and what we think is probably the best way to analyze metagenomic data in the long run, is to analyze everything, to analyze all the data, and not, in essence, chicken out when sequences don't overlap with each other. Because for years, evolutionary biologists and informaticians and others have been working on methods to compare bits of data to each other, even if they don't compare to each other. That is, if you have reference data, you can compare sequences by triangulation to each other. You can ask how far sequence on the left side of a multiple sequence alignment is from reference number 1, and how far is sequence on the right side of the multiple sequence alignment from reference number 1, and try and analyze them in the context of each other, and build a tree of everything from the metagenomic data. And there are a variety of things you might want to do with this. Tom Sharpton, who's a postdoc in Katie Pollard's lab, worked on a method to do classification of OTUs. So this is an important sort of subset of what you might want to do with data. Is this fragment from the same OTU as this fragment, even when they don't align with each other? And you can build, in essence, a phylogenetic tree of all the sequences and try and figure out if they're from the same OTU. And that's what this Phylo-OTU software package that Tom Sharpton developed does. I really wanted him to name this, so it's Phylogenetic Analysis of OTUs. I really wanted him to call this POTUS, President of the United States, but he chose Phylo-OTU. It would have been perfect for election time, too. I'd be happy to talk to people about this on the side. I don't really have time to go into all the details. This is what we did, something like this to try and analyze all of the sequences compared to each other when we did phylogenetic analysis of other protein coding marker genes from environmental data, and found, even for sort of core phylogenetic marker genes like REC-A and RPOB, sequences that did not group into any of the known major lineages of organisms, including bacteria, archaea, eukaryotes, and the known lineages of viruses. So there's data out there that's from really weird phylogenetic lineages. We don't know what those sequences come from, but if we want to, again, compare a fragment that is from a rare organism, and we only have the left half in sample number 1 and only the right half in sample number 2, we again need methods to build phylogenetic trees of all the sequences. And what the real cutting edge in this right now is methods that work like a method called pplacer, which many people may have heard of, developed by Eric Mattson and still developed by Eric Mattson. He's now at the Hutch in Seattle. And there's a software package being developed in my lab by Aaron Darling and Holly Bick, who's here, and Guillaume Jaspin and other people. That's sort of an equivalent to AMPHORA, the package that was developed by Martin Wu, but now trying to analyze all sequences compared to each other rather than just the regions that overlap with each other. And the way these things work is basically what I was outlining. You have a reference sequence alignment. You place individual sequences into the tree of the reference sequence alignment, but you try and place them all into the tree. And then you can even compare the individual environmental sequences to each other to try and figure out if they came from the same lineage or not using certain statistical approaches. And that's sort of one of the things I'm not going to go into detail about, but I'm sure Holly or other people in my lab would be happy to talk to you about. Statistical approaches to analyzing phylogenies have become very big for phylogenetic trees of known organisms. So Bayesian phylogeny, likelihood phylogeny originally, and now Bayesian-based methods. One of the advantages of Bayesian methods and likelihood-based methods is rather than in essence just giving you a tree and telling you that that's the best tree, you get multiple trees. And it gives you a statistical probability of each tree in essence. And so you may not be able to place a sequence into an individual part of the tree perfectly, but you get a probability for where it should go. And that's really important if you want to do things like use a unifrac-like metric to compare 5 different environments to each other. And you get a tree-based metric, which I love, by comparing them to each other. But in essence, you lock the tree when you do that. And you assume that the tree you feed into unifrac is correct. If you instead took a tree that had statistical probabilities of each position in the tree, you could calculate similarities and differences between communities and statistics of those similarities and differences in community based upon the statistics of the tree itself. And that's, I think, where a lot of the phylogenetic methods that people are using to analyze environmental data are going, to take advantage of the statistical approaches that have been used in a lot of the reference organisms or cultured organisms, et cetera, and applying that to environmental data or to very large trees. Like many things in my lab, we're pretty open about stuff, so there's no paper yet published for Phylosift, but the code is out there on GitHub, and Holly and other people even have a blog describing many of the things going on with Phylosift. If you just Google Phylosift, you can find all the stuff about that, and you're welcome to download the software and use it. So one eventual endpoint that I would like to see, but it's pretty hard, is instead of analyzing just one gene at a time, analyze all sequences relative to all genes, and try and, for example, place all sequences in an environmental sample relative to the reference genomes that you have, rather than reference individual genes. And so you could call this method sort of the all in the genome approach to analyzing data. So taking each of these fragments now, so you have fragmentary data from different sequences, and now analyzing multiple genes, if not entire genomes of organisms, and building a phylogenetic tree of everything in a sample relative to everything else in a sample. And to really do this, I've ignored this up until now, but you have to take into account real phylogenetic history. So not just bifurcating vertical evolution, but recombination within species, and that nasty recombination between species, that lateral gene transfer stuff that makes trees somewhat messy. We can actually take that into account from analyzing environmental data. Many people are building phylogenetic networks of reference genomes compared to each other, taking into account gene gain and loss, duplication, recombination, deletion, lateral gene transfer, etc. And now you can overlay environmental data onto that network instead of onto a tree. And in theory, we could place all the environmental data onto that network rather than single one gene at a time. Steve Kembel, again, coming back to Steve Kembel, he's really done some brilliant stuff in metagenomic data, was the first person that I know of to do this in a true phylogenetic context. He basically took the AMPHORA phylogenetic markers, and rather than AMPHORA, which analyzed them one at a time, he concatenated them all together and built a reference phylogeny and then anchored environmental data into that reference phylogeny in this paper that came out last year in PLOS One. I'm not going to go into the details of, it seems to help, at least a little bit with making conclusions based upon environmental data that you weren't able to do if you didn't analyze all the data from across the genomes. So I'm going to shift away from phylotyping now and talk about another example, sort of very briefly, of phylogenetic analysis in the context of environmental data. In lake arrowheads of ancient past, this is what I used to talk about a lot. So the first lake arrowhead that I came to, I talked about what I used to call phylogenomics, which was taking sequences of genes in multi-gene families, or even sequences of genes that just had homologs in other genomes. And when you wanted to predict functions for these genes, what most people did at the time, and still what many people do, is you take the sequence of a gene from your new genome, and you do a blast search or some similarity search of that gene, and you pull out a list of homologs. And one of the problems with doing this is that frequently, when you get a list of homologs, the list has genes with different functions in them. So how do we take that list of genes that has different functions and choose which one to grab and assign to our uncharacterized gene? What I argued at the time, and what I still believe is really useful, is to build phylogenetic trees of the genes, place the new gene in context relative to characterized genes, and use, in essence, evolutionary character state reconstruction methods to infer the likely evolutionary changes in function of those genes over time, which will then allow you to predict the function of unknown genes. Now, I didn't talk about this before, but that's basically what most people were trying to do with phylotyping for many years. Predict the biology of an organism by its phylogenetic placement relative to organisms for which the biology has been characterized. This is the same thing, but now for gene function. So instead of organismal biology overlaid onto the tree, like photosynthesis, chemosynthesis, whatever, we're taking a tree of genes, overlaying experimentally determined functions onto that tree, and predicting the function of uncharacterized genes by the position in the tree. Turns out this actually probably works better than predicting function of organisms based upon ribosomal RNA position. Organisms with the same ribosomal RNA sequence can differ by 40% of their genomes, so it's got some issues with predicting function. But predicting functions from phylogenetic trees of genes actually turns out to work pretty well. You can, of course, do this for any type of data, like environmental data. You can build phylogenetic trees of your environmental sequences, apply the same approaches that you would use for genes from genomes, and try and predict functions of those. The same methods that I just went through for phylotyping can be applied to phylogenetic analysis in the context of functional prediction. So if we want to predict the functions of uncharacterized environmental rhodopsin sequences, proteorhodopsin homologs, we can use those different phylogenetic methods that I described to improve your ability to build a phylogenetic tree and then predict the functions of uncharacterized environmental genes in the same way you would do for characterized environmental genes. So I'm not going to talk about that in detail, but you can apply those same methods. Whether or not it's, of course you should use Phylosift, but I mean, you can use other things. So what I want to talk about just very briefly is the issues with this phylogenetic prediction of functions. So it turns out, if you want to predict functions of genes based upon the similarity of that gene to something in a database, you can do a blast search, or if you're, a snooty phylogeneticist like me, you can build a tree. But what if you do that search and the list of genes that your gene is homologous to, none of them have been experimentally characterized? Who cares about the tree? I mean, it's not going to help you place your sequence in context if nothing's been studied. And so this, of course, is a massive problem. We are generating sequence data at an unbelievable rate. We are not generating functional data at a particularly high rate to go with the sequence data. So we are set with this problem all the time. And actually, first at a Lake Arrowhead meeting, I don't know if it was a talk or just talking to people who were at the meeting, I heard about an alternative approach to doing this, which are non-homology functional prediction methods that were developed by people like Pellegrini and David Eisenberg and Ed Mercott and others. And one of them that we like in particular in my lab is, I'll explain here in a minute, but non-homology functional prediction methods are sort of exactly what they sound like. You're trying to predict the function of a gene based upon some feature of that gene other than its sequence similarity to genes that have been characterized. So a classic example of this is correlated gene expression patterns in microarray experiments. The genes don't have to have any sequence similarity to each other, but if they always show up as being co-expressed under every condition that you look at, you might want to consider that they have some functional similarity to each other. And if you have an uncharacterized gene that shows up as being co-expressed with 75 genes that all have the same function, I'm going to suggest you might want to look at that function that those other genes have, even if there is no sequence similarity. So there's a list of now about 8 or 9 of these non-homology functional prediction approaches. My favorite is something called phylogenetic profiling, where you look at the distribution patterns of genes across organisms. And you group genes by the distribution pattern of that gene, its profile. So you search a gene against genomes and you make a list, yes, no, is that gene and its homologs present across a set of genomes. And you group them by their similarity in their profile. That is, you're looking for co-occurrence patterns of genes. They don't have to have any sequence similarity to each other. We did this many years ago, and I presented it actually at Lake Arrowhead, I think, probably in 2004, ancient history, for an organism called Carboxydothermus hydrogenoformans. It's an interesting thermophile that grows in hot springs off of carbon monoxide gas and produces hydrogen as a byproduct of its metabolism. And we knew that it was probably closely related to sporulating organisms when we sequenced the genome. And then when we annotated the genome, we found homologs of many of the sporulating genes in the genome. People had tried to get it to sporulate previously and it never worked, but Frank Robb was able to manage to get it to form spores and go through the whole endospore germination process. When you look at the genes in the genome and you group them by their distribution pattern of their homologs across other taxa, you see this amazing cluster of genes that are found in all the sporulating species, basically, and not any of the non-sporulating species. And you see many of them are annotated as sporulation proteins, SPO2, SPO3, SPO5, SPO whatever, but many of them are annotated as conserved hypothetical protein. They had never been experimentally characterized in any organism, including the model sporulating organisms like Bacillus subtilis and maybe some of the other Clostridial species. They were there, but no one had ever shown an experimentally determined function for any of them in those organisms. So I'm not going to tell you details about this, but we published a paper showing this, suggesting that many of these genes were involved in sporulation. And actually, 2 days ago, Rich Lozick, who we sent the list to and have been going back and forth for about 5 years now over this list, he's had a postdoc in his lab working on this for a while, and he's actually shown that a huge number of these genes are, in fact, sporulation genes that were missed by biochemical and genetic surveys. I think probably 18 of the 20 that we showed are, as conserved hypothetical proteins, are now known to be sporulating proteins. So this phylogenetic profiling just gets better and better as we get more and more genomes. You know, it would be nice to have more functional information, but we are going to fill in the knowledge of sort of protein pathways across organisms. And if you want to play around with genomes, it's a great thing to do, phylogenetic profiling, to look for conserved functions across taxa. Now you can do this for metagenomic data. It turns out to be much messier and trickier. Susanna Tringe did this many years ago with one of the first sort of metagenomic comparative analysis papers. Ed DeLong's done this in many papers. A lot of people have done this, comparing the presence and absence of genes across metagenomic samples rather than across taxa, and grouping them, again, like phylogenetic profiling, so you could call it metagenomic profiling. It's pretty messy to pull out information from this right now. It doesn't work as well as phylogenetic profiling. We've been collaborating with some people who are more mathematically inclined than I am to try and develop new methods to group genes by their distribution patterns across taxa. There's a new sort of, you could call it a neo-clustering method, non-negative matrix factorization that a bunch of people have been using in a variety of not just bioinformatic applications, but in general in clustering approaches. I can't tell you anything about the math. But we have a paper coming out actually on Tuesday where we've used this with collaborators, Josh Weitz and Jonathan Dushoff, to try and analyze metagenomic data. And it seems to really work quite uniquely compared to other clustering methods. I'm not sure if it's better, but it's different. And it works. So I think it's going to end up being useful for environmental samples. So the last example I want to talk about is using phylogeny sort of in a different way, which many people have heard me, and I'm sure they're really sick of it, talk about in terms of selecting organisms for study. And I've been pretty obsessed with this for many, many years, trying to say, here's a phylogenetic tree of organisms. Here's the list of genomes. They're not coming from a diverse sample of the phylogenetic tree of organisms. And that's been the case since the start of the Genome Project. Some subgroups within the evolutionary tree of life have been sampled pretty well phylogenetically. Actually, the NHGRI has done a pretty good job of targeting diverse vertebrates to compare to humans. There have been many plant projects to sample across the plant tree of life. But for studies of bacteria and archaea, unfortunately, sampling across the tree was not done very well. In the 2010 Lake Arrowhead meeting, I talked about this genomic encyclopedia of bacteria and archaea project that I was sort of coordinating as an outside member of the Joint Genome Institute. Really, the Joint Genome Institute was turning its entire sort of microbial sequencing machine onto this project, and so far we've now sequenced about 250, I don't know if Tanya knows the latest numbers, but 230 genomes from the GEBA project from the phylogenetic diversity of cultured organisms. And it's been this amazing partnership driven mostly by JGI, a partnership with the DSMZ culture collection. But that's you know, that's old news. That's so, can anybody pronounce lambda? I know you're supposed to be able to pronounce some hashtags, but I have no idea how to do this one. But I'm not going to talk about this yet. Maybe Jeff should tell us how to pronounce it. I'm not going to talk about this really in detail today. What I just want to point out is that, you know, genomes are coming out at an astonishing rate, including now good phylogenetic sampling of the diversity of life. How do you keep up? The way I usually keep up is by looking at Genomes Online. Nikos Kirpidis maintains this database. It's genomesonline.org. It's an amazing compendium of all the microbial genome projects that are out there. There's also various databases that track genomes. Morgan Langell, who's been at previous Lake Arrowhead meetings, developed something called MicrobeDB, which is out there in an open source pipeline for keeping track of microbial genomes. What I want to talk about in the context of GEBA is how we can use the information that came from GEBA in essence and other approaches to improve our ability to do phylotyping. So phylotyping works, but it doesn't work exceptionally well in all cases, in particular when lineages have not been sampled well for their genomes. So one thing that we've been doing is going through the genomes that have now been generated with the GEBA project, as well as with other genomes that are out there, and rather than using the 31 phylogenetic markers that we and Per Bork and other people identified from across the diversity of bacteria to sample the phylogenetic diversity of metagenomes, we've been going through each major lineage of archaea and bacteria and identifying phylogenetic markers that seem to be robust for that group, as opposed to for all bacteria at once, or all archaea at once. And you can see, for example, for some lineages, there are hundreds, if not up to 1,000 marker genes that appear to work robustly for doing phylogenetic analysis of that clade. So when we scan through metagenomic data, we can actually place, with sort of robust phylogenetic markers, a much greater fraction of the data than we are currently doing with AMPHORA or these particular phylogenetic marker approaches. To do this, we need a reference tree. We need to have a big phylogenetic tree to anchor all these environmental sequences. That's the picture I took of the tree that Jenna Lang now, sorry. She was Morgan at the last meeting she came to. Jenna Lang has been working on in my lab to build a tree of all the genomes using sort of these new statistical-based approaches to analyzing genomes. That's been submitted and revisions are in the work. I don't think you can see the individual taxa on this tree, but we'd be happy to share it with you if you need a reference tree for genomes. Another thing you can do with all these new genomes that are coming out is improve your ability to make functional predictions from environmental data. So we've been going through all of the genomes that are available, in particular the phylogenetically diverse genomes, and identifying the protein families that are present in those genomes. Now there are great databases of protein families that are out there, whether it's PFAM or TigerFAMs or COGS or a variety of other databases, but they don't get updated very rapidly. And with the speed at which sequence data is getting generated, one thing we need to do is figure out ways to update the protein family databases with new families and adding sequences to the old families rapidly. So we've been working on sort of very rapid Markov chain-based clustering algorithms to take all genomes, all the peptides in all genomes, search them against all other genomes, and identify protein families as rapidly as you can possibly do, although there's no way we can keep up with one high-seq machine anymore. But we'll pretend like we can keep up, at least at some level, and identify protein families in those databases. And Guillaume Jospin in my lab and Tom Sharpton in Katie Pollard's lab and a variety of other people have been working on analyzing these protein families. We've released a data set of all of these protein families. We've built alignments of all the families, hidden Markov models of all the families, and that will allow people to scan through environmental data for more protein families than anybody is looking at currently and get rapid alignments and even trees, if you want them and you want them, for sequences from environmental samples. There's a program that you could use, which I would encourage you to actually use for many of these cases, that Martin Wu developed when he was in my lab. He's now at University of Virginia, called Zorro. It's for masking sequences, so we called it Zorro. Get it? And it basically takes a sequence alignment and will use a probabilistic model of comparison of the sequences in order to identify which regions of the sequence alignment are in essence not statistically supported, and you can mask those out from analyzing, building a phylogenetic tree. And he showed that there are some improvements, in particular with distantly related sequences, that you get when you have this masking as opposed to analyzing a complete sequence alignment. I'm going to skip over that last little thing. And then, so just to wrap up, this, so what I've been trying to talk about in this sort of latest context is how we can use better sampling of gene families or genomes in order to leverage that information to then analyze environmental data. And I think the most striking thing that came from our Genome Encyclopedia paper and from my work on the Genome Encyclopedia project is actually something we could have done even without sequencing any of the genomes, but we hadn't yet sort of wrapped our brains around. If you take the tree of life, let's just take the ribosomal RNA tree from Silva or Green Genes or some database that's out there, and you take that tree and you count the branch length in the tree. That's a metric called PD, or phylogenetic diversity, the sum total length of the branches in that tree. If we take that and we count that for different subgroups of the tree, what we see is shown in this plot. So organism, the number of taxa that we look at on the x-axis, and the sum total branch length in the tree for those taxa on the y-axis. If we do that for our Genome Encyclopedia project, so in blue here are the genomes that were sequenced before our project. They came to a total of about 20 units of PD. We sorted them by the amount of PD that they contain relative to other organisms. So the 18th E. coli genome, sorry all you E. coli fans out there, didn't add a lot of PD to the tree. It added many genes because of lateral gene transfer, but not a lot of ribosomal RNA diversity. Each of our Genome Encyclopedia genomes added a lot of PD. They better have, that's how we picked them. That would have been pretty messed up if they didn't add a lot of branch length, because that's how we selected organisms. If you look in dark gray here, that was all the organisms that were in the Green Genes tree that were described as cultured organisms at the time. Sum total of 180 units of PD in this tree. It's sort of arbitrary units. We would need about 5,000 genome sequences to sample half of that phylogenetic diversity, something to that effect. There's in fact a project going on at JGI right now to start to tackle this by sequencing all the type strains from across the diversity of bacteria and archaea. And eventually we will fill in the PD of cultured organisms. But look at the light gray. That's the uncultured organisms that were just in that database from like 5 years ago, before the sequencing went crazy with Illumina and 454 sequencing, although many of the things coming out of that might not be real. But we would need 10,000 genomes to capture half of the phylogenetic diversity of the uncultured organisms that had full-length ribosomal RNA sequences in the Green Genes database as of 2008, or 2008. That's a massive number of genomes from organisms that we have not yet grown in the laboratory. Technically challenging to generate those genomes. If we actually look at the full diversity of ribosomal RNA that is out there now, the sequences that maybe we trust more than some of the sequences that seem to be error-prone, we probably need hundreds of thousands of genomes to capture half of the PD of bacteria and archaea that are out there. We have sampled nothing. I mean, it's great. We have lots of genomes. They're really interesting. But overall, in terms of the full diversity of bacteria and archaea, and forget about microbial eukaryotes and viruses, we've barely scratched the surface of the diversity of life on the planet. So, building all these protein families and reference trees, it's helpful, but only to a point in terms of what we've sampled. We need projects like, Tanya, are you talking about the uncultured GEBA here? So that Tanya Woike is running at the JGI to go through the uncultured lineages and try and obtain complete genome sequences for those organisms, which are going to be fundamentally important for sampling the diversity of life. Of course, we need experiments across the diversity of life too, so just sequencing things is not enough. But eventually, we will capture some portion of the diversity of microbes that are out there. We will have a phylogeny of them. We sort of have that now. We will have genomes of them. We will have the biogeography of those organisms. We will have the functions of some of those organisms, or many of those organisms. What we're going to have is my dream, as many people know. I used to be a birder. When I first came to Lake Arrowhead, I was actually deciding between multiple jobs, and one of them was not even on microbes at the time, actually. I used to work on birds. I want a field guide to the microbes. And this seemed ludicrous 5 years ago even, 10 years ago. A real field guide where we cover, again, the taxonomy, the phylogeny, the functions, the ranges, the niches, the biogeography, the temporal variation. We can actually start to do this for at least some subsets of environments on the planet for microbes for the first time. So I will leave it at that and thank the many diverse sources of funding that I have obtained for much of the work that I talked about, in particular the Department of Energy for all the GEBA-related stuff, the National Science Foundation and the Gordon and Betty Moore Foundation, as well as DARPA and Homeland Security for much of the informatics work. And then I'll just leave it up there and thank many of the people and hope that you interact with the many people from my group that have driven down to this meeting and probably really want to get out of their chairs right now. So I will thank you.